Welcome to Clin Micro Simplified, where microbiology becomes clear, simple, and a little bit fun. Let's dive into today's topic. In microbial genetics, mosaicism refers to a genome that is not uniform in origin. Instead, it is a patchwork made from DNA pieces acquired from different sources. This concept is extremely important in understanding how bacteria suddenly gain virulence traits, antibiotic resistance, or new survival strategies. Mosaic genomes are a direct result of genetic exchange and evolution in action. A mosaic genome contains regions that clearly did not originate from the same ancestral lineage. These segments differ in GC content, codon usage, or gene organization compared to the core genome. In pathogenic bacteria, such regions often encode virulence factors. Unlike higher organisms, bacteria do not rely only on vertical inheritance from parent to daughter cell. They frequently acquire DNA from unrelated organisms. This happens through horizontal gene transfer mechanisms. When foreign DNA integrates into the chromosome and is retained, the genome becomes mosaic. Over time, multiple such events build a genome that looks like a genetic collage. Transformation allows bacteria to uptake naked DNA from the environment. Transduction uses bacteriophages to transfer genes between bacteria. Conjugation involves plasmid-mediated DNA transfer through direct cell contact. After entry, homologous or site-specific recombination integrates this DNA into the chromosome. Recombination enzymes align incoming DNA with similar sequences in the host genome. This creates hybrid regions where old and new genetic material coexist. Over generations, these regions stabilize and become permanent parts of the genome. This is why mosaicism is not accidental, but an evolutionarily selected feature. Many virulence factors, such as toxins, secretion systems, adhesins, and invasion proteins, are located in genomic regions that show mosaic structure. These regions often resemble pathogenicity islands. Their foreign origin allows pathogens to rapidly acquire complex traits that enhance infectivity without slowly evolving them from scratch. Scientists identify mosaic DNA by comparing base composition and gene patterns. If a region has a different GC percentage or unusual codon preference, it strongly suggests horizontal acquisition. Flanking insertion sequences or integrase genes further support this. Such signatures are textbook indicators used in microbial genomics. Mosaicism allows bacteria to adapt quickly to host defenses, antibiotics, and environmental stress. Instead of waiting for random mutations, bacteria borrow ready-made genetic tools. This explains sudden outbreaks of more virulent or drug-resistant strains and is a major concern in clinical microbiology. Clinically, mosaicism explains why two strains of the same species can differ drastically in virulence. It also explains the emergence of new pathogenic variants. Understanding mosaic genomes helps in vaccine design, diagnostics, and tracking the evolution of infectious diseases. To summarize, mosaicism reflects the dynamic nature of microbial genomes. It arises through horizontal gene transfer and recombination, clusters virulence genes, and gives pathogens an evolutionary edge. Studying mosaicism helps us understand not just where virulence comes from, but how fast it can spread. Thanks for watching. If you understood this topic, make sure to subscribe for more simplified microbiology explained in a clear and simple